The contents of this stack are designed or compiled to assist you getting through the MCAT-style passage, the chemical structure consequences of beta-lactams. Specifically, penicillin. Penicillin is a type of beta-lactam. So this whole passage revolves around the structure of penicillin. A beta-lactam is a blank-membered cyclic ring, or better yet, cyclic amide. So how many parts are in a beta-lactam? All right, it's a 4-membered ring, okay? sp2 hybridization occurs when a carbon atom is attached to blank groups. So how many constituents are bonded to carbon when it's sp2 hybridized? 3. And I might have alluded to this in a previous video. sp2 hybridization means that carbon has one double bond, could be double bonded to another carbon. It could be double bonded to an oxygen, making a carbonyl group, or a nitrogen, yielding an amide group. So it's bonded to one of those, and then typically a hydrogen or another carbon or something else. Uncompetitive inhibition takes place. That's supposed to be place, not places. Ignore the S. Upon the binding of an inhibitor to an E blank dash and S blank complex. So E blank dash S blank complex. That would be the enzyme-substrate complex, okay? So just try to keep uncompetitive, non-competitive, competitive straight, because there's several categories of this type of competition when it comes to enzymatic function. So start to compile a list of them in your head. But with enough exposure to the different scenarios, you'll be able to just nail it. Feedback inhibition is when a product of a pathway self-inhibits steps. Is it earlier or later on in the pathway? What's the answer? That would be earlier on in the pathway. So let's say this little circle or circle-like object is your product. Once you have like sufficiently high concentrations of that, this product can then bind to, I don't know, a hormone or some type of the enzyme that's functioning. It can bind to it and then stop it from functioning or stop it from churning out more of the same product, okay? So it would inhibit steps earlier on, not afterwards, not later on down the road. Blank antagonism is either due to allosteric binding of an inhibitor to an enzyme or permanent active site binding. So what type of antagonism? Blank antagonism. The answer is non-competitive antagonism. That's non-competitive antagonism.